So we'll start off by talking about some of the methods of physical control. So heat is a pretty effective method to kill microorganisms. And there's moist and dry heat. Moist heat is more effective in killing microorganisms than dry heat. And I'll show you a little chart that compares the two in a little bit. But with moist heat, it causes coagulation and denaturation. Coagulation is clumping. Denaturation is the unfolding of proteins. So the microorganisms themselves clot or clump together, and the proteins unfold and become useless. So the microorganism dies at that point. Dry heat removes the water from organisms and causes incineration, but it takes a long time to remove that water to a point where it's actually going to kill the microorganism. So if you look at moist heat, for example, our standard autoclave usually does 121 degrees Celsius, and we usually do it for 15 to 20 minutes, so time to sterilize is 15 minutes. But the same temperature using dry heat would take 600 minutes, so you can see how much more effective moist heat is than dry heat. So there's steam under pressure, non-pressurized steam, pasteurization, and the boiling of water. Steam under pressure, the most common tool that we use is an autoclave. So an autoclave is something we actually have in the back of the classroom. So when we get to our lab period, I'll show you what the autoclave looks like and point that out to you guys. But the autoclave, you put all of your surgical instruments in there. You can put bacterial media. So if we're going to make fresh media for the bacteria to grow, we're going to put it on a tray in the autoclave, and then we seal it up. We fill it with some water. We seal the door tight, and then this whole chamber pressurizes and produces steam from the water that we put in there. And everything on the inside of the autoclave gets sterilized. Now, once you're done, as soon as you open up the door, if this surgical equipment isn't wrapped inside, usually they use like blue colored towels to wrap it up. If it's not wrapped up, as soon as you open up the door, anything in the air is now coming in contact with all of these instruments. So typically we would wrap up these instruments before we put them into the autoclave. That way, when we open the door and stuff settles on the outside, it's settling on the outside of a package, not directly on the instrument that we wish to have sterilized until surgery. Non-pressurized steam, intermittent sterilization or Tyndalization, you expose to free-flowing steam for 30 to 60 minutes, and then you incubate for a day, and then do it again, and then repeat for 3 days in a row. A single exposure is not sufficient to kill all spores, and this is used for some canned foods and certain media preparations, but it's not really commonly done. Pasteurization. So Louis Pasteur came up with the idea of pasteurization. Louis Pasteur liked boiling liquids, and then he liked to see, well, how long does it take after I boil a liquid for microorganisms to grow? Well, through the process of pasteurization, you can't just boil milk. If you've ever boiled milk before, the milk proteins start to break down and the milk turns really weird, and then it tastes really disgusting. So we can't just simply boil it. But through the process of pasteurization, we have these particular methods, and there's a couple different methods that exist, but one of the most common is you heat to 72 degrees Celsius for 15 seconds. So you flash heat it. And that flash heating ends up killing a lot of microorganisms. Campylobacter, Coxiella, Listeria, E. coli, particularly O157 that can eat holes through your intestines and infect internal organs, tuberculosis, paratuberculosis, Salmonella, Yersinia enterococcolytica. So Yersinia pestis causes the plague, but Yersinia enterococcus causes an intestinal infection. So all of these things used to kill a lot of people that drank straight from the udder until we came along with pasteurization, and that made milk a lot safer. One of the other microorganisms that's not listed here is Brucella, and Brucella is still a major public health concern, especially with goat milk. or sheep milk. And people in certain parts of the world consume that type of milk because that's really their only food source. If a mother can't naturally produce breast milk for her children, that's their only option. And due to the area that they live in, pasteurization isn't always easy to come by. So they will drink straight from the teat and end up getting brucellosis, which is a pretty devastating infection. In the United States, this has been eliminated though because we have the brucella vaccine that we give to all of our animals. But again, if you lived in a remote area where vaccination isn't really done or isn't really available, it's still something that can make people sick. Pasteurization does not kill endospores or thermoduric microorganisms. Thermoduric can tolerate high temperatures. They don't like hot temperatures, but they can tolerate hot temperatures. Pasteurized milk is not sterile. You've just decreased microbial numbers. Think about it. If you take a gallon of milk home from the grocery store and you never open it, and you let it sit in your fridge for like a month, it will go bad. Eventually, especially after a month, you're going to see the milk carton kind of bow out from the gas that's produced inside of there from the microorganisms kind of overgrowing. And you may see some clotting of the milk proteins inside of there. But pasteurized milk is not sterile. It just has a reduced number of microorganisms. So with dry heat incineration or a dry oven, the infrared incinerators in our desktops that we use to sterilize our loops for bacterial culture, that's an example of using dry heat. We can also use flames like Bunsen burners, and that's another method of dry heat. And then the dry oven, it's just like your oven at home. So susceptibility of microbes to heat, the thermal death time versus the thermal death point. So this is one of the course competencies, and although I really like to focus this class on medical microbiology, because it's a course competency, we have to talk about the difference between the thermal death time and thermal death point. This predominantly has to do with food processing and food microbiology. But thermal death time is the shortest length of time required to kill all test microorganisms at a particular temperature. So, if I want to kill all microorganisms at boiling temperature, probably wouldn't take too long. But if I wanted to kill all microorganisms at room temperature, 25 degrees Celsius, it would take a really long time because microorganisms can survive in that temperature for a really long time. So the shortest length of time required to kill all test microbes at a specific temperature, you basically pick a temperature and then see how long it takes for everything to die in that culture. The higher the temperature, the shorter the thermal death time. Thermal death point. The lowest temperature required to kill all microbes in a sample in 10 minutes. So now you want everything dead in 10 minutes. So you try to find the lowest temperature where everything would die in 10 minutes. At 25 degrees Celsius, room temperature, you probably wouldn't get everything to die ever in a 10-minute time period. So you would have to start at a minimum of like boiling temperature, 100 degrees Celsius. And at 100 degrees Celsius, you probably couldn't get everything to die in 10 minutes. So you would just keep increasing the temperature to try to find the lowest temperature required to kill all microbes in a sample in 10 minutes. Again, this has to do with food processing. We want to make sure that we can get rid of all microorganisms in a particular food product before we ship it out to be sold to people and consumed by people. Cold. So typically cold usually just slows down microbial growth. So that's why we put food in the refrigerator instead of leaving it out at room temperature, because in the refrigerator, cold slows down the metabolic activity, so it slows down the rate of their enzymatic reactions. It prevents them from multiplying. Desiccation is eliminating the water from a microorganism, and lyophilization is eliminating the water and freezing the microorganism. So with lyophilization, it's like freeze drying the microorganism, while desiccation is just removing the water. Both of these can be rehydrated to become viable microorganisms again. And in fact, lyophilization, that's how our bacterial cultures arrive from our biotech companies. So we go out online and we order up a bunch of pure cultures, and they come in this little tube of powder. And then we put media in with them and put them in the incubator for a couple days, and that's how we bring them back to life. But they were freeze dried when they arrived to us. So lyophilization is really a method of transporting bacteria safely. Two different types of radiation, ionizing and non-ionizing. So ionizing radiation causes ions to form. Non-ionizing radiation does not cause ions to form. Ionizing radiation is used for meat, some types of produce. But the advantage of using ionizing radiation is that it has penetrating power. So if you have a thick cut of steak and you treat it with ionizing radiation, it can penetrate straight through that thick cut of meat. Now some people hear radiation is going on their food and they start to freak out like, oh my God, we're all going to die of radiation poisoning, because people go crazy when they hear certain terms. But it's completely safe, or they wouldn't be doing this to our food. As soon as that beam of exciting ions form, it goes away as soon as you stop the machine. So it is a very self-limited process. Those ions don't remain in the food, and it's very safe to eat. And another advantage of using this is you're not using heat. Like with meat, you don't want it pre-cooked at the grocery store. You want to be able to bring it home and prepare it how you want to prepare it. So it allows us to bring raw cuts of meat home so that we can eat it safely. Non-ionizing radiation, the best example that we would use is ultraviolet radiation, or UV. UV is used in healthcare facilities and research laboratories all the time. It's that blue light that you see illuminated in hospital rooms, surgical rooms, and even in hoods, like what you see pictured here in letter B. What non-ionizing radiation, or UV radiation, does is it causes the formation of pyrimidine dimers. Pyrimidine. Pyrimidine are the bases T and C, thymine and cytosine. So any place that you have a T next to another T, a T next to a C, or a C next to another C, you can cause this cross-linking to form. So here you can see under normal circumstances in double-stranded DNA, you want the A to bind across to the T to bind across to the A, the G to bind across this should be a C here, and this should be a G here. But it should be like a complementary strand that you see going across. But in the case of a pyrimidine dimer, those hydrogen bonds actually form between the two pyrimidines that are next to each other. And that's induced by exposure to UV radiation. Now, if this happens in a bacterial cell, and we try to make a copy of this, and we go in here and try to copy this, as soon as your DNA polymerase 3 hits the T attached to the T, it kind of comes across it and says, well, what is this? And it doesn't really know what base to add. And if this happens to be in an essential area of the genome, this could kill the bacterial cell. And it happens a lot and enough times where it severely mutates their DNA and they end up dying from exposure to UV radiation. This happens to your cells as well. Every time you go out in the sunlight, pyrimidine dimers are forming in the stratum basale layer of the skin. Now the good thing about us is we have many layers of the stratum corneum, that top layer of the skin, and that's all dead. And it acts as a really good barrier for the penetration of UV. The other thing is we have melanin, and melanin is the pigment that is a protein that can fill up our cells. And the more melanin you have, the more resistant you are to UV. So UV can't penetrate through that melanin protein. It's kind of why we tan in the summertime. So you expose your skin to UV radiation and you produce more melanin to try to protect yourself. Unfortunately, people that constantly expose their skin to UV radiation, sometimes when these pyrimidine dimers end up forming, they aren't repaired and they go unnoticed by the enzymes that you have inside of your cells that are supposed to repair these. That leads to the development of skin cancer. Filtration. So HEPA filter, or high efficiency particulate air, that's the common filter that can trap bacteria. So the pores are so small that bacteria can't get through. It's kind of like a coffee filter. So when you make coffee, the coffee grounds are on the top of the filter paper. The water goes through the top and then it goes down into the coffee pot at the bottom. Only the coffee is small enough to go through the filter paper, but the coffee grounds stay on top of the filter paper. Here, the pores are so tiny that bacterial cells won't even go through the filter paper. The problem is viruses can go right through because viruses are much smaller than bacterial cells. And the substance that you're trying to disinfect has to be either a liquid or a gas. And then osmotic pressure. So adding a lot of salt or sugar creates a hypertonic environment, and that messes with the osmotic pressure, or the osmolarity of the solution, which messes with the osmolarity of the bacterial cell. Causes plasmolysis in bacteria, and it makes it impossible for microbes to multiply. Cured meats are treated with high salt concentrations, so they can be kept for long periods of time without refrigeration. High sugar in jams and jellies also has the same effect.